Hello and welcome to the Good Tutors lecture. Today we're going to be looking at the control of gene expression. This is actually just in the middle because I haven't had a tutorial on gene expression and I'm talking about gene expression. This tutorial is actually a response to a request made by some students. Henceforth, in subsequent time, I'm going to do tutorial on the expression of gene. With that said, now today's tutorial we're going to really just be focusing on few points. Firstly, we're going to look at the importance of gene regulation and also tell you what is gene regulation. Next, we're going to look at the levels of gene regulation. So if you have studied the gene expression, you will notice that there are possible levels at which gene can be regulation. Though you were not told, but I'm going to explain or describe those levels to you. Next, we're going to look at the first level, which is changing the structure of the DNA, meaning alteration in the DNA structure. At that point, it is important to note also how gene is regulated. And lastly, we're going to look at the translational control of gene regulation. This is really important. Most of our time is going to be spent on the translational control of gene regulation because the organism we're discussing here, bacterium, uses this level as a central point of regulation of this gene. So thank you so much. If you're not yet subscribed to this channel, please try and do so. Just hit the subscribe button below and you'll be notified every time I post a new tutorial. Thank you. Let's get on. The importance of gene regulation. Firstly, before going on to tell you this importance, let's look at the gap. You've done the central dogma theory, right, of molecular biology. You remember that it states or describes the pathway of information flow from the DNA to the RNA, then to the protein. So if you notice, it will tell you that the DNA gets transcribed into an mRNA and an mRNA gets translated into a protein. However, the central dogma theory doesn't tell us how the information is regulated along this molecular pathway. So you remember that it just tells you DNA is transcribed into RNA, RNA is transcribed into protein. That is how a gene is expressed. That is how our cell or bacterium express gene in their environment. But it doesn't tell us anything of such. However, it is important that this entire information path will be regulated. Let's take, for example, the bacterium that lives in our large intestine as human. This bacterium solely depends on the food that we eat. If you take in glucose, the bacterium will break down glucose. If you took in other simple sugar aside from glucose, the bacterium has to adjust itself to break down that sugar. The reason is the bacterium is trapped in that environment. It cannot leave our large intestine to look for food anywhere. Henceforth, it has to be what? Flexible. So, if you take a keen look at the bacterium, it has genetic information for synthesizing many proteins. That's to include enzymes. But however, only few of those genetic information is expressed at any time. Meaning, like I stated, right? If you have a change in the environment, you have new expression or you have the expression of new genes or proteins that are appropriate to help the bacteria survive that environment are then produced. So if the bacteria is using glucose and the human host doesn't take in glucose, it will make no sense that the bacteria keep producing enzymes that are needed to break down glucose when in fact there is no glucose. So all of this is made possible because of regulation of this gene. So, in summary, for unicellular organism, it allows for this flexibility. I mean, you are able to, or it is able to switch off one pathway and then turns on another pathway. However, how is gene regulation important to humans? This actually back down to the process of doing embryological development, right? One concept is all of the cells in our body have the same gene. But however, during embryological development, we have genes being switched off based on the function of cells. So let's take for example, right? The neurons in our body is responsible for the transmission of impulse. So if there is a gene that this neuron had for, breaking down toxic substances like liver cell or transporting oxygen as in the blood, the neuron doesn't need that gene. It is not part of its function. Henceforth, those genes will be or deactivated or silent in a neuron, and the function that it is adapted to, the gene responsible for that function will be activated. So, for multicellular specialization, gene regulation is very, very much the important. So there you have it. That is why we have to regulate the expression of gene. Let's get on. So we've now told you why it's important to regulate gene expression. So where are the levels at which we can regulate the expression of gene, especially along the information pathway, the central dogma, as we will call it. So the first level here is alteration of our DNA or chromatin structure. This is actually important when it comes to eukaryotic cells because prokaryotic cells, the DNA is not really compact as the eukaryotic cell, which happen because we have or we need the DNA to fit into the nucleus. The next level of regulation is transcriptional control. Next, we have the processing level. After transcription, the mRNA is processed before being sent to the cytoplasm. So at that level, also we can carry out regulation. Also, mRNA stability. When the mRNA gets into the cytoplasm, the stability of the mRNA actually regulates the expression of the DNA, meaning it will tell us whether the RNA will stay long in the cytoplasm to be able to produce more protein or they will get degraded faster. And number five here, we have the translational control. And lastly, we have the post-translational control. So there are so many levels of regulation. All of these levels will really make more sense when we're talking about eukaryotic gene regulation. However, for prokaryotic gene regulation, the central point of regulation is the transcriptional control. In this tutorial, we're going to highlight the alteration in the DNA structure that accounts for control of gene expression and the translational control. So let's move on. The first level here is the alteration in the DNA structure. So one thing you need to know is there are proteins that bind to DNA sequence and affect the expression of a particular DNA sequence. Those proteins are generally termed as DNA binding proteins. So the DNA binding proteins are capable of binding to a particular segment of the DNA strand, and by binding to that segment, they will regulate the expression of that particular segment. So the DNA binding protein have functional parts that are called the domain of that particular protein, and it's approximately 60 to 90 amino acid. So In a domain, only few amino acids commonly interact with the DNA. So, though the domain is made of several, like I stated, you can see the number of amino acids, but you only have few, and those few are usually asparagine, glutamine, glycine, lysine, and arginine. So, those are the common domain or common amino acids that actually interact with the DNA, and it is actually based on some part based on the polarity, right? Yeah. So that's one reason that it counts for these amino acid interacting with the DNA. So the DNA binding protein usually binds temporarily to DNA molecule. So they don't just bind to the DNA molecule and bam, stick there forever. So they bound and what? Release the DNA molecule. Bound and then release the DNA molecule. So this actually allows for competition with other molecules that are involved in carry out the process of transcribing the DNA. So with that, now we'll move on to look at other aspect of this regulation, which is the types of DNA binding proteins. There are different types of DNA binding proteins. So the types are actually grouped based on the characteristic structures of those proteins known as the motif formed at the binding domain of the DNA. So the motif are simply structures, right, such as the helices that fit into the major groove of the DNA double helix. So you remember the minor groove and the major groove of the DNA, right? So the motifs are actually structure that enable the DNA binding protein to be able to fit into the major groove of the DNA. So these are some few examples. There are a lot of them. The first term here, we have the helix term, helix motif, which is actually two alpha helices that bounce to the DNA major groove. The next one, we have the zinc fingers motif, which has to do a loop of amino acid, right? And zinc at the base. So, and lastly, we have the leucine zipper, which has to do with a helix of leucine that binds to adjacent major groove. So these are examples of some DNA binding proteins that actually bind to the structure of the DNA and change the conformation of the DNA, either protecting the expression of a particular gene. Next, next we look at translational control. So you understand that the alteration in the DNA structure is actually done by regulatory proteins known as DNA binding proteins. So one thing you need to remember is there are certain factors that allow for the expression of a particular gene when it comes to the DNA structure. So you've often heard about the methylation and the histone acetylation. Those are two major concepts that are actually important when it comes to DNA expression. Sorry, I didn't include that in the slide here, but methylation actually will inhibit the expression of gene, and meaning it allows the DNA to remain compact to the histone protein, whereas the acetylation of the histone protein in the DNA will allow the DNA to uncoil, thereby favoring the process of translation. There is also different structures of DNA, right? Like the euchromatic DNA and the heterochromatic DNA, right? The heterochromatic DNA structure doesn't allow for the process of gene expression. So you have the DNA tightly packed around the histone proteins, whereas in the euchromatic DNA structure, you have the DNA freely attached to the histone protein, which favors the process of translation. So I just thought to highlight those processes since I didn't in fact include them in the slides here. So now let's look at the transcriptional control. One of the major points at which, like I stated, we have regulation of bacterial gene expression is the transcriptional control. And this is usually done through operons. So have you heard that? Operons as stated. So operons are a set of structural genes. So when it says structural genes, as I will elaborate further, they are actually genes that code for proteins. So the genes that are responsible for the coding for proteins, these are the genes we refer to as structural genes. So an operon is a set of structural genes, one, that could be two, three, four genes that have a single promoter and they are transcribed as a unit producing a single mRNA. So the mRNA that is produced from an operon will serve as a template for the production of several proteins. Don't worry about the fancy terms like the promoter and so forth. We're going to break all of those terms down in subsequent slides. So, like you see here, we have regulatory elements. Regulatory element in the structure of the DNA, they are just DNA sequences that are not transcribed, but they affect the transcription of other DNA sequences. So you're going to see a lot of regulatory elements. Example of one such regulatory element is the promoter or the operator. So this segment of the DNA, they are not transcribed. However, they regulate the expression of other segments of the DNA. Let's move on. So the definition of our terms continue. Next, you have the regulatory genes. So the regulatory genes are usually a sequence of the DNA that will code for proteins or RNA. And what happens is the proteins that are coded for will interact with the DNA sequence and affect the transcription of that particular DNA sequence. So the regulatory genes usually produce proteins that are termed the regulatory protein. So the regulatory protein will either serve as a repressor, meaning it will stop the expression of a particular gene, or it will serve as an inducer and stimulate the expression of a particular gene. That is the importance of regulatory gene. Don't forget about this. Structural genes, like I stated, they are the sequence of the DNA that encode for proteins that have specific function in different types of metabolism, biosynthesis. So you have several proteins as we will discuss in different types of operons. So the structural genes are of different categories. There are some genes that actually are regulated, meaning those genes are always on because the cell needs them to carry out its daily function. So those genes are generally called the housekeeping gene and they are expressed continuously. Or we could call them the constitutive gene. Don't forget about that. So constituted gene, in summary, they are genes that are expressed continuously without regulation. So you've seen an expression or an exception of a gene that could be expressed without we worried about the process of, oh, how is it regulated? So let's get on. Okay, so let's have a picture of what we've been defining and what we've been discussing. That's an operon. So if you notice, like I stated earlier, An operon is a group of structural genes plus their regulatory elements. So this is an operon, as you can see here. Firstly, the operon has a single promoter, and then look here, there is an operator. And thirdly, you have gene A, you have gene B, and you have gene C. So all of these components combined together constitute what we call an operon. So firstly here you have the promoter. You remember the promoter serves as the binding site for RNA polymerase. So RNA polymerase binds to the promoter and carry out the process of transcription. Next you look here is the operator. The operator serves as the binding site for a regulatory protein. So how is the regulatory protein formed? You remember I told you that there is a structure of a regulatory gene that has its own promoter. So RNA binds to the promoter of this regulatory gene, carries out transcription, and then the mRNA is produced and translated into a regulatory protein. The regulatory protein now goes on to bind to the operator and either inhibit the process of transcription or facilitate transcription. That's the importance of this entire segment of the operon. Next, you have gene A, B, and C. These are the structural genes. These genes code for proteins or enzymes that are important in different metabolic pathways. So once the RNA polymerase binds to the promoter and the environment is feasible for the transcription of this gene, you have the gene being transcribed into an mRNA. The mRNA gets translated into different proteins or enzymes, and these enzymes go on to function in different metabolic pathways. So this is just a great schematic of the definitions that we've had and also help you understand what really an operon is and those components that I usually discuss. So let's move on. So upon understanding the various aspects of an operon, what it is, let's look at the types of translational control. There are two broad types of translational control. We have the negative control and we have the positive control. The negative control actually is a process that will stop gene expression. Hence, the regulatory protein serves as a repressor. So what do we mean? Mostly you have the transcription process on, but if there is a production of the regulatory protein, that regulatory protein is going to serve as a repressor. That's one way for. The next one is a positive control. Generally, this is a control that stimulates the expression of a gene. Hence, the regulatory protein is going to serve as what? An activator. So a simple way to really remember this, I would tell someone is to think of positive as something that allows the process to go on, and negative, no, right? It stops the process from going on. That's just the simple way I thought to remember this. So the next thing is operons can also be classified as inducible or repressible. So inducible operon is an operon that is normally off and sometimes it is turned on, whereas the repressible operon is an operon that it is normally on, but whenever we have the presence of a regulatory protein or a factor, that particular transitional process goes off. So that's as simple as it is. With that now, let's move on. So let's look at the negative control of gene expression. The negative control here, it could either be inducible. Like I said, in the types of translational control, first we said there is a positive control and a negative control. Next we said the control could also be inducible or repressible. So those two categories can be combined, as you can see here. So we have negative inducible operon. So what do we mean? For this type of operon, normally in the absence of an activator protein, we have a regulator protein here that is usually active. So the structural gene produces a regulator protein that is active, and this regulator protein binds to the operator and prevents the binding of the RNA polymerase. So once RNA polymerase cannot bind to the promoter, it means that this gene cannot be transcribed. But look down here, we have an inducer present. So once an inducer is present, it binds to what? To the allosteric site of this regulatory protein. So once it binds to the allosteric site of the regulatory protein, it prevents it from binding to the operator. Henceforth, the RNA polymerase can now bind to our promoter and carry out the process of transcription. So this is a negative inducible control. Like you said, on negative control, what happens? A negative control usually turns off the presence of the regulatory protein, what turns off the transcription of this gene. But whenever you have an inducer, what happens? It inactivates the regulatory protein, and then we are able to have the transcription of our gene. So hope is as simple as it can, because I try my best to really break it down for you. Let's move on. The next one is the negative repressible control. So we are combining the names, right? First, we have negative inducible, negative repressible. So negative repressible here, the regulatory protein that is produced is normally inactive. So this means that the regulatory protein cannot bind to the operator. Henceforth, transcription is normally on, as you can see here. So the transcription is on and we have our proteins being produced. However, when we have the production of this protein, there is something known as a feedback loop, meaning we have accumulation of this product. This product now serves as what? An inactivator or repressor, meaning they bound to our regulatory protein and actually prevent or enable them to bind to the operator, thereby preventing RNA polymerase from binding to the promoter. So you see this, right? This is just a simple analogy of what I've been telling you about the regulation of gene control. So we are producing a product here that has accumulated in the cell. Because we don't want to keep wasting resources, this product is going to bind to the what? Regulatory protein and cause it to become active. So once the regulatory protein becomes active, it binds to the operator and the RNA polymerase cannot bind to the promoter to facilitate transcription. So it is a negative control, however, it is repressible. So negative control in the presence of an active regulatory protein, what happens? Transcription is off. Don't forget about that for a negative control. In the presence of an active regulatory protein, transcription is off. But for positive control, in the presence of an active regulatory protein translation is on. Don't forget about that. It's so simple. So the next step of translational control we're going to look at is a positive control. So in the positive control, as I stated, when you have an active regulatory protein, it's going to work. It's going to stimulate the process of transcription. Whereas in negative control, if you have an active regulatory protein, what happens? It stops the process of transcription. So a common example we can think of is in a lactoperon. Lactoperon, there is a protein known as the catabolite activator protein. The catabolite activator protein actually binds to the promoter and increase the efficiency of RNA polymerase. So what happens is in the absence of this protein, there is this sloppiness of the RNA polymerase, meaning it doesn't really sticks to the promoter, it bounds and then falls off. However, with the presence of this catabolite activator protein, it helps in the efficient binding of the RNA polymerase, thereby facilitating the process of transcription. Let's move on for you to see better what I mean. So, before discussing a type of operon, it makes more sense if we first understand how lactose is metabolized in the cell. So, firstly, we have extracellular accumulation of lactose. There is a transporter here that is known as permease. So, permease allow the transport of lactose into the cell. So when lactose get into the cell, there are two possible pathways. And these possible pathways are actually facilitated by an enzyme called beta-galactosidase. So beta-galactosidase will either break down lactose to produce galactose and glucose, which is then for further use in the cell to produce energy. However, it also relates to the production or conversion of lactose to allolactose. The allolactose serves as the inducible agent or the inducer of the entire lactoperon. Allolactose can then further be converted to galactose. So, remember this entire pathway and actually get a grasp of the normal metabolism of lactose in the cell. This will actually help you understand how the lactoperon works. With that, now let's move on. So we look at the lactoperon negative inducible operon. What does this mean? From our previous discussion of a negative inducible operon, it means that when we have an active regulatory protein, transcription will be switched. You see it? We have a negative inducible operon. Negative operon, we have active regulatory protein transcription as well. So what happens is, in the presence of lactose, you have the production of what? Allolactose, right? So allolactose will bind to the regulatory protein that is transcribed from the regulatory gene that is somehow upstream of our normal gene. So you see it? This is the regulatory gene. It's on the same DNA strand, but it is far from what? Our gene of interest. So when this is produced and we have lactose present in the cell, allolactose bound to this and makes the once upon a time active regulatory protein inactive. So once lactose binds to this and this become inactive, what happens? The protein can no longer bind to the operator, meaning that RNA polymerase is now free to bind to the promoter and facilitate the process of transcription. So in the lactoperon, there are three genes, as you can see up here. First, we have LAZ, LAC-Y, and LAC-E. These genes are transcribed into mRNA that code for three different enzymes. So the first enzyme here is the beta galactosidase. You remember beta galactosidase, sorry. As you remember, that was responsible for the breakdown of galactose. Sorry, did I just say galactose? What's going on? Responsible for the breakdown of lactose to produce glucose and galactose, right? Next, you see the permease here. Remember the normal metabolism of lactose. The permease facilitate the transport of lactose into the cell. And we have the last one, which is transacetylase, which the function is not really that known. So this become a positive feedback, right? Whenever you have more production of galactosidase and you have more allolactose being produced or more permease, permease is going to facilitate more transport of lactose into the cell. Is it clear? Let me go over this, right? So when we have the production of these enzymes, the more we produce permease, permease facilitates the entry of lactose into the cell. And you have more galactose or galactosidase present to break down the lactose into what? Into galactose and glucose. So the more product we have, the more lactose we incorporate into the cell. This is just how it works. So this will go until we have deficiency of lactose or reduced lactose in the cell. Then this entire pathway will be slowed down. The next control we're going to look at is the positive control, which is the catabolic repressor. So in bacterium, glucose is mostly preferred as compared to lactose. So they have so many reasons for that, right? One reason is it takes less energy to break down glucose as compared to lactose. So the cell sometimes try to save energy, and with that, it's going to prefer using more glucose as compared to lactose. So when glucose is present in a bacteria, the process of lactose or lactoperon is actually turned off, except you have deficiency of glucose and you have lactose present, then you can move on to activate that entire process. So aside from lactose, the metabolism of other other sugars are turned off in the presence of glucose. Remember that. And this is called uh, catabolic repression, meaning because you have glucose present, which is further being broken down, is going to cause the inactivation of other pathway. So the process of glucose inactivating other pathway is what we term as catabolic repression. So the translation of the lactoperon is absent when you have glucose present and lactose absent. So the catabolic repression actually it works in the format of a positive control. Despite the lactoperon suggested a negative control. So what does that mean? Let me break it down, right? So this catabolic repression actually it occurs in addition to the negative control that we just discussed, meaning the lactoperon that we just discussed. So this process will go on when there is low lactose in the cell. So the positive control is achieved in a sense that when there is no catabolite activator protein, the binding of RNA polymerase to the promoter is not really that efficient. So the cap binding protein will bind to a site that is upstream the promoter. When you hear about upstream, downstream, it's mostly the point at which or the point towards the promoter is termed as the upstream or the start point, whereas the point towards the gene or the last structural gene is termed as downstream. So right before the promoter, you have a site that facilitates the binding of the cap protein. So once cap protein binds to the DNA, is going to facilitate the binding of RNA polymerase. However, how does this happen? First, the cap protein will form a complex with cyclic AMP before binding to DNA. So when you have low glucose in the cell, you have high level of cyclic AMP. So the cyclic AMP comes about because you have the breakdown of more ATP into AMP, and the cyclic form of the AMP is more stable or is more common as compared to the other structures. So once you have low glucose, it means that the energy level will be low, so we have high level of cyclic AMP. So once you have high level of cyclic AMP, they are able to bind to cap protein and bind to the promoter of the transition or the structural gene. However, when you have high glucose in the cell, the CAM level becomes low. Hence, once there is low CAM level, the possibility of the cyclic AMP binding to the CAM complex is actually low. Henceforth, the process of RNA polymerase binding to DNA or the DNA to facilitate translation becomes very much inefficient. So that's how the cap repression actually goes on. So in the sense of positive control when there is presence of the regulatory molecule or when a regulatory protein is active, what it does is it facilitates translation. So when a cap protein is active, what happens? It facilitates the process of translation. It enhances the process of translation. And when it's low or inactive, it what? It doesn't support translation, but rather turns off or slows down the process of translation. That's just as simple as I can place it. So this is what I've just been discussing here. So you have the cap, where you have high level of cyclic AMP. The cyclic AMP is going to bind to the cap protein and form a complex. This complex is going to bind to a segment upstream. So you see here what I was discussing about upstream. Here's the promoter, and these are the structural gene. Up, the promoter is where we call the upstream, right? So the CAM complex binds to the upstream segment of upstream side of the promoter and facilitates the binding of RNA polymerase. So once this is present, RNA polymerase binds efficiently to the promoter and carries out the process of translation, and we are able to get our needed products here. However, when there is low cyclic AMP, the possibility of cyclic AMP binding to cap become less. Henceforth, the cap cannot bind to the to this regulatory site. Hence, RNA polymerase also cannot bind to the promoter. Therefore, you cannot have translation, or translation will be reduced. That is just the simplest way I can place this. Thank you. We've come to the control or the end of this tutorial. This was actually just based on the lack operon. So in subsequent tutorial, we're going to look at the trip operon and also look at other levels of gene regulation. I hope this tutorial was helpful, and I'll see you in the next. Don't forget to subscribe and share with friends. Thank you.